I'm glad that all worked well, audio-wise. So I have the pleasure now of introducing our final plenary speaker. So we will next be hearing from Dr. Jay Brands. He's a professor at the Skidaway Institute of Oceanography, the University of Georgia, and the director of the Skidaway Island Scientific Stable Isotope Lab. He received BS degrees in both chemistry and oceanography from Humboldt State University in California, and a PhD in chemical oceanography from the University of Washington. Dr. Brands is a marine and freshwater chemist who studies carbon and nutrient cycling in the environment, as well as marine debris and microplastic contamination in Georgia estuaries and nearshore waters. He does this by leveraging the tool of stable isotopes, which allow his team to determine sources, pathways, and rates. So with that, I'd like to welcome to the stage Dr. Jay Brands.
So just checking to make sure that this thing works, because I have a tendency to wander around a bit when I talk. Thank you all for coming on the final day. Thank you again, Paul and Rebecca, for inviting me to this. It's a distinct honor to be able to speak to you all. And, this is an interesting subject. Now, she talked about stable isotopes, and I did that most of my career, but in the last 15 years or so, I became more and more interested and concerned about this whole field of microplastics, which everybody hears about, and most of the time you hear about it through articles in the press. And I'm going to talk a little bit about how maybe we can try and understand this material in a little better way, but we still have a tremendous amount of things to figure out. So, microplastics are perhaps the most confounding material that I have worked with in the organic chemistry and carbon cycling world. They have a really unique level of complexity in terms of nearly ubiquitous amounts of sources in our environment through every possible pathway you can think of. They have an incredibly wide range of chemical compositions and physical properties to them. It becomes incredibly complex in terms of even talking about one particular category. When you talk about polyethylene, for example, when you talk to a chemical engineer, it's immediately like, okay, well, which version of polyethylene? Low molecular weight, medium molecular weight, high molecular weight, ultra high molecular weight, and all sorts of different additives and dyes and plasticizers. And each one of those will modify the parameters of things like breakdown susceptibility to photochemistry, susceptibility to biological breakdown, weathering, physical and chemical. They have a wide range of densities as a result of this, and that gets manipulated for different types of products and materials. They're making things for a specific consumer product, and they can modify it in many different ways, which don't necessarily show up, especially when you're looking at these things with your instrumentation. They have an extremely wide range of sizes. They're not necessarily manufactured in this fashion, but they break down into smaller and smaller pieces. And I think what is not necessarily well understood at this point is the wide range that these things are extending in. Everything from sub-micron down into the tens of nanometer size range, all the way up to floating docks that are meters in scale. And we don't call those microplastics, but it's still a continuum of plastic materials in the environment with different sources. We also have a wide range of morphologies on top of that, and I'm going to try and make the case today that that's an important parameter for us as scientists to consider. They're persistent, wide range of susceptibilities to all these different breakdown pathways, and therefore you can have materials that have lives in the environment from scales of weeks and months to essentially thousands of years, depending upon not just their structure, but the type of environment that they are located in, whether it's sea surface, whether it's deep sea or sediments. It makes a huge difference. And finally, a demonstrated ability, and this is the one that gets the public really concerned about this and interested in this subject, a demonstrated ability to cross biological barriers and be transported up food chains. So the idea that you are what you eat, well, when you eat something from the store and it's wrapped in plastic, it's been demonstrated that some of that plastic can get into you. It has also been demonstrated that some of that plastic can get into your bloodstream and get into your organs and get into your brain. So there's this whole world of concern that's coming out, but there's also a whole world of just not understanding what's driving all of this plastic in our environments. And there's finally a wide range of interactions with other pollutants. They are certain types anyway, kind of become effectively contaminant sponges. On top of this, they're darned hard to measure, at least to measure in the types of parameters that really fully inform you about their composition. There is no simple or complex instrumental method that is out there that can provide rapid measurements of size and composition at the same time, and distributions across a full range of microplastics. This is especially true for the really small stuff. We have a real dearth of information out there that can give us the structure of these really tiny particles. Even within size ranges, there are a number of different methodological, this has kind of become I guess it's grown from the ground up rather than being driven from top down design. And so everybody has their own way of doing things, which in the beginning was good because we were starting to generate data. It was very egalitarian, but it does end up making it really, really hard to do intercomparisons, to do summaries of, let's say, for global budgets and so on. And we've seen some talks on this during this very meeting. So again, at present, there's no sampling routine, no sample prep, sample size range, instrumental technique, or intercalibration standards that have been agreed upon by the scientific community. So we're still very much in our infancy in this field. Okay, but it's an infancy that's had a very, very long incubation period. This meeting turns out to be exactly 2 weeks after the 53rd anniversary of the first, to my knowledge, underreporting of microplastics in the marine environment. This is done by Ed Carpenter and Ken Smith. And they noted microplastics in their plankton toes in the Sargasso Sea. These were exactly what we would think of as microplastics today, even though they didn't use that particular terminology. Later on, Ed reported that he found polystyrene spherules, of the sort of basic building blocks of polystyrene materials, in Narragansett Bay. And he furthermore followed up with a contact of his in the chemical manufacturing industry. He wished to thank Ed Harding for notifying all polystyrene manufacturers about this contaminant in coastal waters, because he was concerned about it for a variety of reasons. As far as I can tell, there was no response recorded. I mean, he didn't publish a follow-up to this. And this warning kind of was lost to the field for 30 years. In the meantime, chemical manufacturing has gone exponential. And you will see this in other talks during this meeting, and you'll see it a lot, that we have an exponential growth of the production of plastics worldwide. It's rising faster than the growth of humans. And it is noteworthy that even when you look into this data, you find that there's enough variability in there that the actual number is hard to come by. And this is the starting point for all of our other estimates. This is the number that we should have the best. It gets worse when you start talking about amounts of plastic in the oceans, and it is far worse when you start talking about numbers of particles. Now, in this talk, I'm mostly concerned about numbers of particles because the concentration of microplastics, or we think about as a concentration proxy, is going to be set by the number of particles per liter. And estimates have ranged from 5 trillion in global aquatic environments in 2014. It was very quickly bumped up to 50 trillion, and it's kind of stayed in that area for a while. We have now numbers, and the most recent numbers are something like 80 to 300 trillion, except for this one outlier up there at the top, 120 million trillion. So we were talking about orders and orders of magnitude higher. Now that estimate was basically kind of just a theoretical exercise of what happens if all the plastics that we have put into the environment become microplastics and stay in the environment. So clearly a very much upper bounds of things. But I am going to argue today that we are nowhere near understanding the number of microplastics in the environment, because that very small fraction, which may be a significant portion of what's out there, we have very little information on. And I'm also going to make a point that I made 19 years ago at this very meeting, that small stuff is out there. This is data from a study that I did with Ron Benner at South Carolina, looking at North Sea organic matter that he collected with ultrafiltration. So he's trying to collect really small stuff. This is the particulate side, not the dissolved side of ultrafiltration. But I was looking at it using a technique called scanning transmission X-ray microscopy. And essentially, this instrument has a resolution of 50 nanometers, and it gives you the bonding environment of carbon in your samples at that kind of resolution. And in these samples, the first time I'd ever seen this, and I'd been looking at other samples from the rest of the world, I started to see these really odd spectra. And the odd spectra had these bumps in this area and this area. There was no natural organic matter that had ever shown that to me. But in looking at the spectral database, it started to show up, okay, well, that can be a reflection or can be explained by Teflon. And there were other plastics that showed up very clearly that also had odd spectral features. Unfortunately, X-ray doesn't show up things like polyethylene and polypropylene very well, because they look like sort of natural organic matter. It doesn't have NMR specific level of information. But the smaller pieces on here were down to the microscope's resolution limit of 50 nanometers. This stuff is out there. Okay, so let's return to how do we start getting a handle on this sort of thing. I know there's been a lot of kind of depressing news lately. And one of my hopes is that we can tackle this field in a more defined manner, and we can do it with the help of our fellow citizens, our non-scientist citizens in here. Microplastics by their nature are labor-intensive to study and instrumentally time-consuming to study. And this is particularly true in the estuarine systems that I do most of my work in. We have a helper in this, and that is the public. The public is very interested in microplastics, as you can imagine. They see these stories in the paper. They want to know more. They want to study their own backyards, their own ponds, their own river systems, and so on. And so you'll have people that will come and say, how do we do this? How can I have, an exercise in my science class in 6th grade? How can we look at microplastics? How can we do this? The NGOs that are running the River Keepers programs are like, we would like to monitor our river systems for microplastics. Now, how do we do this? And so you can use these folks as force multipliers. You have to be cautious about it, though, because they have a lot of enthusiasm, but it has to be done in the right way. So you have to keep your methods simple, cheap, and available as much as possible if we want to harness this enthusiasm. And so basically, when you're studying my type of environment, which you have a Secchi disc of less than a meter depth, you can barely see your hand when you stick it under the water in this type of environment, you have a needle in a haystack problem. You might have to filter a liter of water in a very pristine environment to pick up 1 or 2 microplastic particles. So you have to clean it up. Now, other environments, you don't have this problem as much. But I recommend that you use the least harsh chemical pretreatments together with the fewest isolation concentration steps. You do have to concentrate them. Typically, you can't just look at a liter of Mickey water and see, oh, there's a microplastic in it. But if you concentrate them and clean it up a bit, then they start to appear. However, you have to be careful because it has been shown that every time you mess around with a sample, every time that you transform it or concentrate it, you run the risk of both losing microplastics, especially these small ones, and contaminating the system, because we are surrounded by microplastics. If I were to filter air in this room during this particular talk, I would pick up microplastics that are airborne in this environment. Go ahead and, there we go. And what is really on top of this, anybody that has worked in the system has seen this. They are incredibly spatially variable. So this is a series of samples from the area around Savannah, Georgia, where I work, and you have orders of magnitude difference in concentrations of microplastics within a mile spatial distance. And there's not a lot of rhyme or reason other than in general the concentrations are higher around the cities, but not tremendously higher. You still find microplastics in other places that visually look pristine and yet still have a ton of them in there. But you have to be able to capture these hot spots. You can't just go to one spot, take a sample, and say that, okay, this is what we're seeing in the environment. And the problem gets worse because those spots change. So you get hot moments, this concept from the river hydrology field. And this is a series of samples that were taken at one of those hot spots that was taken over about a 1.5-month period at low and high tide. And this was a summer intern project that studied this. And again, order of magnitude differences in different tides, but not every single tide. You couldn't say, oh, all high tides are high or all low tides are high or whatever. It was very chaotic in this system. And we've run this experiment a couple different times, and we always get this kind of chaotic process. So you have to sample a lot in order to follow this sort of thing. This also has implications for things like, okay, if you want to understand what a filter feeder is experiencing, what level of contamination, if you don't do this type of temporal sampling and you just do it once, you're not going to necessarily capture what they are seeing. And also, if you're doing experiments, you're not necessarily going to capture what they are, whether they're seeing a chronic level or whether they're seeing an episodic level. So citizen science can harness the public interest in this field, but the studies need to be designed around their limitations. They need training because these samples are easily contaminated. They need sample processing to be kept simple. Typically, we have our citizen scientists do the preliminary size fractionations on this. And we do identification as quick and inexpensive as we can at a certain level. And I'm going to turn around and say that that is not sufficient, but it is at least something that gets them in the game, and it also gets you some preliminary estimates of what might be in your samples. And to that, I'm going to put a little plug. I wish that I were talking earlier and we hadn't already had the poster things, but I'm working with Dr. Sue Ebanks and James Bird at Savannah State and trying to use inexpensive light sources such as 365 UV handheld flashlights and 400 UV nanometer blue light flashlights, as well as visual light, looking at these samples under a regular old dissection scope or a relatively low magnification scope, and materials in those samples will fluoresce at different wavelengths. So we're doing a kind of a cheap and dirty hyperspectral analysis of this, but we're using our eyes, which are really pretty good at looking at different colors that are being emitted by these. And you can get people into the game with these. They're $20 to $30 for each set of light sources that you're using on here. And the microscopes can be found in a lot of high schools and other places, and even in some grade schools if they're well funded. And so it's a very nice teaching tool because you can shine something on there. Let's say all of a sudden you're looking at a mass of brown stuff and it's hard to tell what something is. You shine the UV light on there and all of a sudden something glows bright red, and that gets their attention. And you can make tallies of these things, and you can say, okay, well, it glows bright red in this wavelength and maybe doesn't glow at this other wavelength, and it looks like this in visual light, and gather all of these things up. What James has been doing is building a flow chart of this type of information. So you can say, okay, what color does it look like under this, this, and this? What do these things mean? What do these pieces of information mean? What do they suggest that is? Some of the things that fluoresce are natural, and you can identify those and reduce your false positives within your samples. So this is nice and cheap and easy, and you can do it. And I have done these types of things with grade school students in Georgia, and it really gets them excited and makes them understand the joy of both learning the scientific process and understanding what we're doing in the environment. But my warning here is this, that is not sufficient. And while it is great and it gets people excited and it does provide you a kind of 30,000-foot view of what's in your samples, there is no substitute, if you are serious about understanding microplastics in your environment, for not taking a subset of those samples, as many as you can afford the time and energy to do, and look at them under instruments that can tell you exactly what that material is. It was a very eye-opening bit of work that I did with Barbara Beckingham and at College of Charleston. They had a collection of white fibers that they had collected from the stomachs of fish that they were collecting up in the South Atlantic Bight region. Every fiber had passed what they call a hot needle test. And this is an early test that was used to try and identify plastic materials. And the idea was, if you have a material and you put a red hot needle, touch it to it, if it melts, that must mean it's plastic. If it chars, that must mean it's not plastic. And that was a standard method that was kind of developed early on, about 15 years ago, in this field, and a lot of people used it. So they had gone through and done this test to all these different fibers, but they, of course, wanted to know what kind of microplastic it was. So they sent me hundreds of these things. And I sat there during COVID and analyzed them on a confocal Raman microscope, which we had just recently gotten installed at Skidaway. And I called them up and said, about half of these things are cotton. Did you know that? And they were totally shocked. Now, you look at them under the microscope, under 50x, 100x microscopes, and they still, a lot of them did not have features. Now, they were probably all manufactured cotton materials, but they were cellulose-based. And the only way you could tell the difference between these things was with a technique like FTIR or Raman that told you the bonding environment of it. And they would show up very clearly as having very different, Raman spectra. So again, it is really critical, if you want to understand what's in your samples, to use these types of techniques and hopefully develop cheaper and more rapid analyses, because these types of Raman analyses take at least 5 minutes on a perfectly isolated sample and can take up to a half hour on a sample that does not give you a nice spectrum immediately. Okay, but when you do this sort of thing, at least in my environment, so this is coastal Georgia, estuarine waters outside of Savannah, you find that something like 70% of the microplastics that you look at are of a fiber morphology, more than 10 to 1 length to width. And of those fibers, the non-cellulose fraction is dominated by polyester variants. And if you look at this graph, well, these last two bottom ones are polyester and polyethylene phthalate, and they're like 90% of the composition of this. This was another summer study by another intern. We see little bits and pieces of all sorts of other microplastics in there and other plastic and polymeric materials, but it's dominated by this. So this starts to have implications. What we're looking at is an estuarine system that appears to be dominated by textile-based microplastics based on polyester. That sounds like something that, we might wear. And our working hypothesis is that in the estuarine, open estuarine waters proper, this is what survives all the different winnowing processes. And why do I think this? Well, I can take advantage of other people that have worked in the field. Joel Weinstein has done yeoman's work at the Citadel over the last decade, and he's been looking at these things too. And he also sees in the open water areas around Charleston Bay that he's seen 70% microplastics fibers, not other morphologies. But if he looks closer in, let's say the source rivers that are really close into Charleston, which are really intimately in contact with the different communities there, that he sees much lower proportions of fibers in those and much higher proportions of other morphologies, and he sees tire wear particles primarily right next to the roadways. They're not getting very far. Furthermore, where are these things going if we have these other morphologies in there? Are they just non-existent? And that's not true. There's a ton of non-fiber morphologies in the sediments. They are sedimenting out into the salt marshes in this area. So what we're seeing is kind of a size exclusion chromatography model. It's not really exclusion, but it's a size trapping morphology type of model in this system, where you start off with everything, and as it moves through this environment, different fractions are collected, and what you're left with are the fibers in the open waters. Those are the ones that are going to interact with the shrimp and other organisms that are living in those waters. So I can take Liz Canuel's estuarine sea cycle model, or estuarine, and she did a very nice job, and I'm just going to clumsily put my plastic morphology hypothesis on here. Let me see. We can get that. Yep. That the densest materials, tire wear tends to fall in the densest fraction, sediment out very quickly, even in high energy, and these areas have 2-meter diurnal tides. There's a lot of tidal energy in them. And less dense particulate matter is trapped out in marshes, and it's not density, because a lot of the particulate matter is polyester. It's the same type of plastic that we see in the water column, but it's a different morphology, and it is something about the particulate morphology that gets trapped in salt marshes. And then finally, the low density fiber morphologies, because we don't see a lot of the high density fibers out there. We see mostly polyethylene and polypropylene out into the water column proper. Well, if you want to do something about that, you start to tell the public, hey, if you really want to control what's getting out into your estuaries proper, stop purchasing plastic materials that will get out there, and that means textiles. So I always tell people, if they just start being more conscious about what they buy, that if manufacturers make stuff and it doesn't get taken care of, it doesn't get bought up, they will stop doing it. This is the way the system works. So we just had some implications on this, that composition, density, and shape are important. Morphology can provide clues as to source and possible mitigation strategies. And the microplastic concern for whatever ecosystem you are studying is really going to depend upon what's being trapped in that particular environment, or what is escaping to that particular environment. And with the last few minutes of this, I'm going to give you I don't know if it's a humorous story or it's a cautionary tale. So microplastics, you can put as much energy and effort as you can spare into a study and still be completely surprised about the results. And I've seen this more often, not just at my work, but with results that are presented at meetings and talked about with students and talked about with lay people. You can lay out a really beautiful experimental system and be completely surprised. So consider this. 5 years ago, we had the shutdown, and it was an opportunity. I had a new student that had come in that was very interested in studying our aquarium system for microplastics. It's a very small system. We're fortunate to have it on our main campus. It has about 7,000 square feet of space, 16 different types of, or 50 different saltwater tanks, including a touch tank where people can come in and grab stuff and the kids can look at stuff up close. And an average of about 45,000 visitors a year pre-COVID. Well, March 2020, we all remember that the world shut down. And this included the aquarium. It shut down. No more visitors, no more open doors, nothing. They only had the very minimum number of people in there to take care of the animals and maintain things and sweep and that sort of stuff. So it remained that way until around late May 2022. And so you can see this very clearly in the visitor log. So I'm going to have to go back here because I'm not hitting the right button. We have a long period with only roughly 5 to 10 visitors a day in there. And then this sudden shoot up to about 200 when everybody was getting out. Everybody wanted to go do stuff. So we have an interesting situation. We have some reasonable hypotheses that we can make. This increase in visitors, we know it's starting off at a low level. This increase in visitors should see an increase in microplastic contamination. We've seen other studies and we've seen from our own experience that people are microplastic sources and they will contaminate your samples if you're not really careful. So this is a reasonable supposition for us. Second of all, they took the time during the close down that they were going to remove all the carpets from the public areas and replace them, which is a smart thing for them to do. But there's a disturbance event. So we thought, okay, if we capture before, during, and after, we will see that carpet renovation. Carpets are made of nylon. We double checked this. We thought we'd see a burst of nylon in the system. And then finally, that touch tank with the highest surface volume ratio and the most exposure to visitors would be the one that was contaminated the most. And we thought these were very reasonable hypotheses to test. It's almost like, oh, well, these should be trivial. So we picked a series of tanks. Oops, I'm gonna go back here, keep hitting the wrong button. This is the touch tank up here, a couple of tanks in the main aquarium space, and then the source water reservoir, or source water comes from the Skidaway River and goes in there and is settled and filtered. So we wanted to see if we were gonna see changes in the tanks versus the source water and changes in the touch tank versus the others. Okay, then these were triplicate sampled in each one of these, and my student went to heroic efforts to make sure that we had 0 blanks in our samples, and then they were triple filtered, 35 nanometer, 106 nanometer, and 63, not nanometer, micron sieves within this to try to separate things out and reduce the complexity. And then they were pre-treated with 25% hydrogen peroxide and heated to 60 degrees, which is a very gentle way in the grand scheme of things to remove most organic matter. We did this to try to not lose as many microplastics as we were trying to retain as many as we could. And then they were finally filtered onto 0.08 micron polycarbonate filters, and every single particle under the microscope was examined under Raman. We were not trying to be selective at all. And because of that, it would take my student 2 weeks to go through every single one of those filters. So she couldn't up the number of samples during the process, but she did for every 2 weeks, all the way through basically a full year of looking at these samples. Okay, so this is kind of hammering it as hard as we can in terms of understanding this nice controlled ecosystem. So the first thing is that we found, and this is not a surprise, that a lot of what we had left in these systems was cellulosic, organic, quote unquote, non-plastic. And we divided that out, that's that orange fraction. The other colors represent microplastics that we identified by composition. There are a few of them that even the Raman couldn't quite figure out, so we've left those as unknowns. But interestingly enough, in this system, it's not polyethylene that's the dominant fraction, it's polypropylene in that light blue that is the dominant fraction. So we're not looking at just simply a mirror of the external world. during the lockdown. So these are box and whisker plots, and the first one on the left is just total particulates. The one on the right is that little pie section of identifiable microplastics. And what we saw was, okay, we're not seeing a lot of variability in the tanks, which would imply to us that we're not picking up a lot of stuff inside, which all made sense to us at the time. We saw a higher level of particulates in the touch tank, but not microplastics. So this would imply that dust and clothing fibers and so on were the main source of contamination, airborne contamination in this system. Okay, so we're okay with this for now. Open condition, and this is where things kind of slapped us in the face that we didn't understand this system at all. We saw significant differences in microplastic abundances, the one on the right hand here, between the reservoir and other tanks, but they went in the opposite direction than we thought. They were lower in the external tanks, and they were lowest in the touch tank. This did not make any sense, and my poor graduate student came to me within the first 2 sampling events after they opened up and said, "There's something wrong. I don't see any microplastics in my samples. I don't know what's going on." And we went over everything that she was doing. She hadn't changed a thing in her sampling. They hadn't changed a thing at the aquarium. They just opened the doors. Furthermore, if we look at time series, first of all, you can see the variability in particles. Oops, let me go back. There we go. Particles, and in total particles and in microplastics. And this red line right here represents when it opened up. You can see almost a step function in there. You also see, interestingly enough, that there were peaks when the carpets were changed. So we did see something there, but those peaks were not nylon. It wasn't the nylon that was coming out of this particular exchange. It was other stuff, and it was mostly non-plastic materials. And finally, when we summarize this data and we look and try to understand things in more detail, I'm going to keep hitting the wrong button. There, up here, total particles, and they went down. Microplastics were significantly down. Everything with a red star is a significant decrease. One of the big ones that dropped was that polypropylene fraction. That's what's driving that whole drop in there, and we don't really understand why that is at this point, but that was the one that was the big driver. Polyethylene dropped, but the only one that rose were Teflon-like materials. And those rose, but they're so rare in the system that they couldn't drive the kind of big signals that we were seeing. And it's not like people are wearing Teflon clothes or anything like that, but it was in there. Everything else, there was no significant difference. So, some reasonable hypotheses, but unreasonable data. All 3 of them were disproven. Increase in visitors would cause an increase in microplastics. No, we saw a decrease. Carpet removal would cause an increase in nylon. No, we didn't see that either. And finally, a touch tank with that highest ratio, contamination levels are the lowest, but there was really not a huge difference between the touch tank itself and the other display tanks. It was almost a step function in everything. So finally, possible reworking of hypotheses that carpet acted as a particle sink, that perhaps it was so full of dust and other things in spite of being vacuum that when they removed it out and put a new carpet in, it acted as a new particle sink. It's also been suggested that the system was more or better ventilated because you had a lot of visitors coming in and exchanging the room air with the outdoors, which would say that the outdoor environment is cleaner than the indoor environment, and that's not necessarily unreasonable. So some basic things about this: we always need more data. We need to use force multipliers to try to do it, and we always have to double check with high resolution chemical resolution techniques. You can't consider microplastics as one contaminant, even though we like to summarize this as, you know, even in that case, summarize them in categories. It's not one. They behave differently. Each type has its own story to tell. And so we have to understand these both from size, shape, and composition in order to try to get a better handle on things. And also, all this variability is really implying that we are missing some essential drivers in this field. I have not a rational explanation for this at this point, but everybody sees it in their fields in a whole bunch of different types of ecosystems. They see this intrinsic variability of this particular contaminant, and it's something that we are going to have to figure out as a scientific community. Finally, as we've seen with other things, it takes a team to tackle microplastic research, as well as we saw with the awardee here. And as part of that whole thing, I'd also like to make a plea. I have a opening for a graduate student, for a PhD level graduate student in my lab. If anybody has any talented undergrads that are looking to move on into this field and deal with all of this craziness, it is an excellent opportunity, I feel. And with that, I'll say, I got 30 seconds for a question. Anybody? Yes?
Yeah, I have two comments. I expected you to say that they had installed filters at the time after COVID, and it made me wonder if people would, where they have filters, have looked for microplastics there. So that's one comment. Yeah. The other is, I wonder if some of the advantages of the ability to form plastics with specific properties could be used to try to absorb and remove contaminants from the environment. I'm wondering if somebody could develop a plastic that the fluoride compounds, for example, would bind to, and that we might be able to use them for removal.
There are people working on that. Let's take the second part first. There are definitely companies that are working on this and have shown effective techniques at removing a certain level of microplastics from waterways, and they tend to do it more by hydrophobic interactions. They tend to use that type of absorption to it. So there are some things, but of course, they haven't been scaled up to the type of scale where we have contaminated the heck out of our environment. But certainly you could use something like that outside of a potential contaminant point where you suspect it's coming in and trap it out of there. The second thing is, we thought about the filter issue, and they had not changed a darn thing. And when they had a regular filter change that they were doing, but they didn't add extra room air filters. They didn't feel like they were big enough to worry about that during COVID. Now, I don't know about the effect of personal filters on this sort of thing, but we just didn't see effects of that. Question?
Yeah, so I have two questions. So first of all, my impression is that a big source of microplastics is washing machines. When you wash these synthetics, and that generates a lot of microplastics. And my first question is that impression correct? And my second question is then, would it be possible to filter the water from these washing machines in homes to kind of stop the generation of microplastics at the source?
Yeah, you are correct. There are companies that actually sell filters for your washing machines, and it's a lot more of a pain in the rear to deal with for the public than just changing it off of your dryer. It has been well established that a washing machine can generate hundreds of thousands of microfibers and microparticles per wash. And it is becoming more recognized in the wastewater treatment field that they need to design their treatment facilities to try and capture as much of this as they can, but that takes money and time and effort, and all of these things tend to be in short supply, especially these days. I think I need to go though, unfortunately, but I'm happy to talk to you, anybody afterwards.